other times we didn't get much time to do this, the microscopes as well. But today we do, so we'll incorporate the microscopes today. Huh?
They're going to be microscope slides on the test.
No. But we'll study them though. Okay. We'll maximize our learning potential, Elizabeth.
Can't wait.
Right? See how Scott is excited? He's like jumping with enthusiasm. Okay. All right. All right. So urinary system. What is that? Well, by the time I'm done talking about these slides, after about 15-20 minutes, when I say, what is that, all of you will yell out the answers and you will know.
Is it a kidney?
Okay, Brianna, come here. Don't be lazy. Okay. Yeah, it's a kidney. It's a cross-section, and it's a cross-section of the tubules. Okay? Angelique, you can see from there?
Yeah.
Eagle eyes. All right. Okay, so here's a kidney now, okay? And this is a cross-section of about 40x. And you're looking at this section here, okay? This is the renal medulla. And this part here is the renal cortex, okay? Well, gee, how do we differentiate that? How do we separate the cortex from the medulla? Okay, color. Color can fool you sometimes. Okay, so what else?
The cortex and the medulla.
The white part is white.
The corpus.
Yes.
Yeah,
Say it. Corpuscles. What Kadia said. In the cortex, you have renal corpuscles, which are the glomeruli and the Bowman's capsule. Right? Do you have that in the medulla? No. That's your key marker. So in the cortex, you have the corpuscles, okay? And in the medulla, you don't. And also in the medulla, when we zoom in, okay, you will see we have a lot of these, in longitudinal sections, you have a lot of these empty white lines, these spaces. These are the tubules. You have a lot of tubules running longitudinally, which are the collecting ducts and the loops of Henle. Okay, we'll look at that later. And also in the dividing region in between the renal medulla and the cortex, remember from the diagram, what artery was running around the pyramid of the medulla that was separating the cortex from the medulla?
Arcuate.
Arcuate. So here you see the arcuate artery, and here's the arcuate vein. Okay? So that is running here. Interlobar just coincidentally is in this region. Okay? But arcuate, definitely you will use that as a guide. It will demarcate the cortex from the medulla. Are we clear? Okay. Now we zoom in on this. Okay. Now you can see 100x. Okay. Now you see the renal corpuscles. Okay. And you see some distal convoluted tubules and proximal convoluted tubules, and you see glomeruli. At this point, I don't expect you to differentiate at this resolution whether it's PCT or it's DCT. Okay. Distal convoluted or proximal convoluted. However, when we zoom in, okay, now you should be able to tell some structures apart. For example, this is the renal corpuscle, this entire structure, okay, but inside it you have the glomerulus. This is the glomerulus, okay, and here's the lumen of the Bowman's capsule or the glomerular capsule, okay, where the filtrate urine collects, okay. Now next to it you have the proximal convoluted tubule and you have a distal convoluted tubules, because remember they're in the same region, they fold inwards next to the glomerulus, okay, they fold and they come together and the loop of Henle goes down towards the medulla, right? So how will we differentiate now proximal convoluted from distal convoluted? At this point you can differentiate. What do you see different about the PCT and the distal DCT?
The DCT is like
DCT is whiter. What is whiter?
The
The lumen. The lumen is whiter. Okay. Now the question is, why is the lumen of the DCT whiter, and why? Well, then the question is, why is the that means it's empty. There's nothing in the lumen. But why is the lumen of the PCT pinkish? What is the function of the PCT? Remember the function? Reabsorption. In order to do reabsorption, you need to increase surface area. What do you use to increase surface area? Lots of? Huh? Microvilli. There are lots of microvilli in the lumen of the PCT, which will make the lumen of the PCT more pinkish. You won't find these microvilli, lots of them, in the DCT. Make sense? Okay.
Question
Yeah.
about the renal corpuscle.
Corpuscle.
It's the glomerulus and the Bowman's
Yes.
capsule that make it up?
Yep.
Okay.
Okay. All right. Now we're looking at the kidney medulla longitudinal section. By the way, going back to this slide, the cell types in the glomerulus was what? Fenestrated capillary endothelium, which is a squamous cell. Okay. But I want you to write fenestrated capillary endothelium when I ask you for what kind of cell is it. Okay. The capsule is formed of simple squamous. Now, this PCT and the DCT, what cell types are they? What cuboidal? You have to be simple. Can it be stratified? No. There's no room to be stratified. Okay. All right. Now, this is a cross section of which part of the kidney? Medulla or cortex? How do you know it's the medulla? No. You know it's the medulla because you see a lot of collecting ducts and loops of Henle. Well, these hollow tubes that run lengthwise, longitudinally, okay, these are the collecting ducts that run in the medulla. Okay. What are the collecting ducts? They collect urine from all the nephrons, and they run down and they take it down to the tip of the pyramid at the papillae, and urine exits because all these collecting ducts come together. Okay, so you have collecting ducts, and you also have loops of Henle. Well, how would you differentiate those? Go ahead.
That's what I was going to
Oh, that's what you were going to say. Okay, good question. So Kadia is asking, how will we differentiate the collecting ducts from the loops of Henle? Well, what do you notice, Kadia?
The space.
Okay, collecting ducts have wide, have a big lumen. What are the loops of Henle are here in this region. It's
The dark.
they're darker, right? You see these structures here. They look more dark. What color? Reddish, darker reddish.
So these would be the same thing here? Would it be?
Now these here are mostly. Some of these could be yes.
Yeah.
Like right here in this region. Yes. Okay. Yeah. Now these mostly okay are darker, and the loops of Henle are very thin at this point, thinner than the collecting duct. They have a wider lumen, and this region is darker with the loops of Henle. So why is that? And you know enough, Mariam, to answer that question. Why are the region of the loops of Henle, why is it darker? Monique. Avions. Why are the loops of Henle on the histology section? Why is that area of the loops of Henle, why is that darker?
Is that where they concentrate the urine?
No, not urine. Brianna. Nicole. Sandra.
Have to do with like what's reabsorbed or
No, has nothing to do with what's being reabsorbed. The structure.
although
Structure.
Is the cell type different?
You're getting closer. What do we have present around the loops of Henle? When I drew it on the board, especially the loops of Henle going down to the juxtamedullary nephrons, what do they have? Think. Huh?
The capillaries?
Capillaries. It took a long time to get there.
I don't recall. I have to look at that again.
So there are capillaries around the loops of Henle. And what are they called? Sandeep? Lisa?
Huh?
What are the capillaries around loops of Henle called? Miriam.
Drawing
Where's
line.
Miriam?
Right.
Okay. No? No? Jeffrey.
Professor, if any one of us knew, we would have shouted.
Okay. Vasa recta.
Okay. I do
See?
remember that.
I was waiting for this moment. Aha, I knew that. Okay. Vasa recta. Vasa recta means vessels, straight vessels, okay? Because they run parallel to the loops of Henle. And these blood vessels, these capillaries, they run around the loops of Henle. So in whichever regions you have loops of Henle, those capillaries make that histological tissue region darker. Make sense?
But they're around there for what purpose? For exchange of...
Yes.
Okay.
Yeah. Okay. Now we took that region, okay, the loops of Henle, and we are zooming in on this now. Okay. So here you see the loop of Henle, right here. Okay. This is running, and you see blood vessels running alongside it. So how do you know which one is the blood vessel? The pinkish one. Okay, one with the pinkish lumen. That is the blood vessel. Okay. That has RBCs in it. Okay. And the thin lumen, that is the loop of Henle. And adjacent to it, you have the collecting ducts. Collecting ducts obviously have a much wider lumen. Okay?
The capillaries, though,
they're not that visible. Yeah.
Okay.
Yeah. So I wouldn't exactly like say that's a capillary. In this one, not really, no.
Just distinguish between the light pink, which is the
but I
loops
expect
of
you to
Henle.
know that loops of Henle are present, right?
Right.
Because you see those capillaries. But it will be hard for you to pick the capillary out in this, yeah. That I give you. Okay. Now, we're looking at a 1000x resolution of glomerulus. Yes. And the cortex, yes. And you have the glomerulus. So it must be the cortex, if it's the glomerulus, okay? And obviously it's the kidney, right? And that was a joke. Okay. See, I told you, don't expect any major improvements. Okay, you have to suffer until the end. Now, the epithelium now around the glomerulus is what? Which is simple squamous. See this right here. You see these cells? Well, they're flattened, but you see their nuclei. See the nuclei are flattened as well? This is a simple squamous of the glomerular capsule. Everyone sees it?
Yeah.
Okay. And now here, these cells, okay, these are the nuclei of the glomerulus. Which nuclei of which type of cell?
Fenestrated capillary endothelium.
Yeah. Now we have another kind of cell called the podocytes, but don't worry about it for now.
Fenestrated what?
Capillary
Capillary. Sorry.
endothelium. Okay. So at which point are the blood afferent arteriole and efferent arteriole coming in and out? Well, you can't. I don't expect you to know from this one which one is afferent, efferent, but they're both coming in and out from here, this region, right?
Mm-hmm.
Okay. All right. So now we have a cross section, and we can see PCTs, and we see DCTs and some collecting ducts. So here now we're zoomed in on the PCT, and what do you see inside the lumen of the PCT? You see this? What are these pinkish structures?
The microvilli.
Microvilli. Okay. So in PCT, you will find an abundance of microvilli and mitochondria. In the simple cuboidal epithelium, you have two things which are high: microvilli and mitochondria. Well, why is that? Well, again, for the same reason: secretion and reabsorption. The majority of the reabsorption takes place in the PCT, and you spend a lot of energy in those processes, which is why you have a lot of mitochondria as well in the cells of the PCT. And what are the cells of the PCT? Simple cuboidal, right? Okay. So in the DCT, you don't see that, okay? And in this one, you have an adjacent collecting duct, okay? And the lumen is slightly bigger than the distal convoluted tubules. That's how you can differentiate that.
It's also wider here, look.
Wider, yeah. Question? Okay. Now here you again see cross section, and you see all of these are what type of ducts? PCT, proximal convoluted tubules. So you have, this is the epithelium simple cuboidal, this is the nucleus, these are the microvilli, and this is the lumen. Okay, those are the individual structures. And what are the next two? Well, this is what you can see, part of the glomerulus over here. Okay. Now we're looking at the cross section. Is it the cortex or the medulla? Is the medulla. Because now you see lots of collecting ducts, okay, and all the epithelium in all these collecting ducts is what?